All right, good morning. This is actually my second time doing this. Hopefully it goes better than the first time. A little bit new to making recorded lectures, but we'll try again. Yesterday I tried on PowerPoint. You guys saw that it really didn't work, so we're trying a different software today that can be uploaded to YouTube, and I think everyone should be able to have access to it. So again, these are completely optional. You don't need to use these. You could just scroll down to the bottom of Google Classroom and use all of the PowerPoints up there to do your fill-in notes. So if you want a recorded lecture, this is it. Since I am new to this, if you actually have, like, Miss Lockman, you talk too fast, you say a lot, what's going on? Please let me know so I can get better as, the next week and a half go by. And so you have a better experience at these, I get better at these, maybe we'll do a thing. It's all good. Anyway, so let's get into it. So, science starters, I'm still going to have them do when you come back from our social distancing. So please make sure you're doing them. Please make sure you go back and do science starter number 1 if you missed it yesterday. And for today, we'll do science starter number 2. So our 3 questions for today are define diffusion and describe the difference between active transport and diffusion, and what type of cell transport is occurring in the image right here. So diffusion, facilitated diffusion, active transport, what's going on right here? And just give me one reason why you think that. Maybe we should do like a group class chat, and that'll be the question of the day. I'll think on that. I'm still adjusting how we're doing things, so maybe next week we'll make the science starters like a classroom discussion. I think maybe that'll be a little more interactive and a little easier. And plus you can say hi to people. So if you need a moment to do those questions, hey, you could always hit pause, which you can't do in the classroom on me. So that must be nice. Go ahead and pause and answer those. Just a note on the agenda, we are doing the notes right now, but we're not doing this drawing or this cell membrane coloring. Just the only homework you have today is the POGL, which is already up on Google Classroom. Some of you guys have already done it. Kudos. It is not due Friday. It is due today. So that's the only thing we're doing today besides these notes. And they're not too bad. They're pretty short. So let's get through them. There we go. Cell plasma membrane. So how does the cell membrane maintain homeostasis for the cell? How does it maintain that internal environment to be the same no matter what? Like our bodies like to be about 98 degrees. The cell is also like a fairly constant temperature and salinity, like how many, the concentration of salt and all kinds of conditions. So we're going to talk about how the cell membrane actually maintains this for the cell and how it allows things to enter the cell and allows things to leave the cell. So I think you guys have seen this before, but just so you know, the things to keep in mind on this are, this is a protein. We're going to talk about them in a little bit. The actual full name for these is a phospholipid bilayer. So the number you think of when you look at the word bilayer is 2, like by 2. So there's 2 levels to this. You got this first level of phospholipids and a second level of phospholipids, and they're tail to tail. So their hydrophobic tails are facing each other. Now hydrophobic, phobic as in phobias, it doesn't like water, water. So it actually repels water, while this hydrophilic head attracts water. And this dynamic, this play off each other, actually leads to how the entire cell membrane works and how passive transport works. Proteins are when you get into the more active transport, things that require energy from the cell, but the hydrophilic head versus the hydrophobic head play off each other is how you get passive transport. So homeostasis is a balanced internal conditions of cells. You can see kind of things bounce. Some things are bouncing off and some things are actually allowed into the cell. This is outside the cell, and then the darker blue is inside the cell. And it's maintained by a cell membrane controlling what enters or leaves the cell. So these are your proteins. And you can see some things are allowed through and some things are not. So the cell, sometimes it's known as the plasma membrane. So if you're doing any like the POGIL today or any worksheets or readings in the next week, we're gonna have to come. If it says plasma membrane, it's the exact same thing. Don't let it confuse you. So the function, it protects the cell. You know, our skin protects us. The cell's membrane protects it. It's semi-permeable, also kind of like our skin. And we've all had wrinkly fingers from, you know, being in the salt water too much. And so it's also, the cell membrane is also semi-permeable. Some things are allowed in and some things are allowed out. It allows cell recognition and it allows it to interact with the environment. So it actually kind of is allowed to, makes it be pulled from place to place. So we'll go down to those in a moment. The main thing is it maintains homeostasis. It maintains that normalized internal states. So normal temperatures, normal like salts, normal ions, all of that. And that's, you know, homeostasis is the name of the unit, so kind of an important idea. So that phospholipid bilayer, again, there's 2 layers of the phospholipids. Bilayer, 2 layers. So a phospholipid itself contains 2 fatty acid chains that are nonpolar. That's where you get the 2 tails. So it doesn't have 1 fatty acid chain, it has 2. So when you look and see a little picture, just know that each of those is a fatty acid chain. And these are nonpolar. These are your hydrophobic parts of your phospholipid. These do not like water. And your polar head, hydrophilic, it attracts water. Water is a polar molecule. And polar, light goes to light. That's actually a big thing in chemistry. So water is like polar things are attracted to other polar things, and nonpolar things are attracted to nonpolar things. That's why oil and water doesn't mix very well. That's why oil will float on top of water. And that fatty acid tail creates that hydrophobic center in the phospholipid bilayer. So this center inside the layer is hydrophobic because you got tails on tails. You got tails facing each other. Tails on tails on tails. So, a couple notes on the proteins and we'll be good. So, membrane proteins are embedded in that lipid layer. So, any protein embedded in that phospholipid layer, this outer layer of the cell, sorry, that's my dishwasher. They're called membrane proteins. That's kind of an overarching name for them. So, integral proteins extend through both sides of the membrane. These allow for that active transport. So there'll be an opening on either end. Transport channel proteins aids in moving substances across the membrane. Receptor proteins bind to substances outside the cell. This is kind of a lock and key kind of thing. So if this substance has the right key, it'll fit into the right lock and they'll hook together. While this transport channel, these guys, aids in moving substances across the membrane. And the shape of the substance, its chemical structure, is actually a big deal when you're transporting this stuff across the phospholipid bilayer. It's kind of like when you were a kid and you had those toys where you had to put the square peg in the square hole and the round peg in the round hole. If you try to put the square peg in the round hole, it's just not going to happen unless you really take that little mallet you got with it to it. And then you're breaking the cell layer and then it's all bad. So just think of it that way. You have to have the right kind of shape and you have to have the right kind of structure to really use those. Glycoproteins with carbs, these are for cell identification. So this is what these like weird hexagon things are kind of going on about. This we'll probably get into tomorrow, if not Monday. All right. So membrane proteins, part 2. Peripheral proteins are only on one side of the membrane. So these have a very specialized function. You're definitely not going to see them as much. But that's if like this part didn't exist, it just kind of got cut off right here and it was only open to one side of the membrane, or even the other way around. And this is where enzymes come into play. Again, we're going to do a deep dive into all that fun stuff later on, but right now we're going to focus on that vocab, getting all the words down so you even know what I'm talking about. So those enzymes assist in chemical reactions. So it takes what would be a normal, like a curve on a graph, and really skyrockets the process. It takes a normal kind of chemical reaction rate and really sends it through the roof as far as how the amount of chemical reactions that are even happening. That's the point of the enzyme. Enzymes play a pretty big role in cell membrane structure and processes. You don't really see them a lot in the body because usually you don't like to skyrocket what's going on in your body. You don't want to all of a sudden heat up all at once. So enzymes, they have very specific jobs, and we'll talk about a few of those the coming days. So a lot of people talk about cholesterol as being bad for you, and you can't have too much cholesterol, but it's really used in that bilayer for strength. It actually forms the strength of your cell membranes. So it is really important to, have some in your diet. Of course you don't want to overdo it, but that's really what it's used for. I'll post this when I post this video. And yeah, that's about it for today. I hope you got your notes. Please do your POGIL, and I will, if you have any questions, log into the office hour at 10:30, and I will see you guys tomorrow.